All right, so moving on to lecture 3, probably this lecture will be a review for most of you guys. We're going to talk about some genetics behind blood bank, so on the molecular level. And again, this should align with what you guys are discussing in molecular as well. But we're going to talk about how some of these genes are inherited, and that determines what antigen is going to be expressed on the cell. So you guys all probably know that what blood type you are. It's all hereditary based on what genes are present, depending upon the specific allele at the specific loci, all right, will determine what sugar is added onto the cell membrane, which will determine what type you are, okay? So it's all hereditary. So not only for the ABO group, but also as we start moving into other blood antigens, when we start looking at RH group, we start looking at the KEL group, the Duffy, the KID family, okay, they are all hereditary. That one chromosome is passed on from each parent, okay? And that is why sometimes you can use, well, you guys all know you can use ABO determinations to determine paternity testing, to rule out a father, possibly. And so that is why genetics plays such a huge role in that. So when we do our blood bank testing, we are actually doing what we call serological testing. When we do blood bank testing, we're actually looking for we can tell the phenotype. If you think about a patient's ABO, unless they are AB, meaning that they have both the A and the B antigen being expressed, or they're O, meaning that they're amorph, right? They don't have either the A or the B antigen being expressed. We don't really know what the genetic ABO determination of that patient is. We know what their phenotype is. We don't necessarily know what the genotype is, right? Because if you're an A, you could be homozygous, have the A allele expressed twice, or you could be AO, all right? So that genetic makeup, we don't know just based off of our serological testing. The only way to determine that, we can make inferences, all right? And we can predict what their offspring might be, but the only way we could confirm that would be to do a genetic or a molecular genotype, okay, on the molecular level. So that's the kind of difference between our serological testing. Now, when we start getting into further antigens, like our antigens are all grouped into families based on their inheritance pattern, based on their reaction characteristics and things like that. When we start getting into like the Duffy family, you'll have haplotypes that are inherited together. So you have Duffy A and Duffy B, they're inherited as a pair. So you're either Duffy A positive, Duffy B negative, Duffy A positive, Duffy B positive, or you're Duffy A negative, Duffy B positive, okay? So they're always gonna be inherited together as a haplotype. And so they're kind of grouped in a family. And as we start moving into those antigens, that'll make more sense to you. And so you have the Duffy family, you have the KID family, you have the RH family, you have the M, N, and S system, and that we'll talk about in a little bit. Okay, so those are all inherited as haplotypes. They're inherited together. And it's all based on the genetic level, what is present on the chromosomes, how that DNA is synthesized for protein synthesis. All right, and you guys all know this, we all know this basic cell biology. Each cell of the body has a nucleus. Inside that nucleus, that's where your genetic material is. We're looking at the chromosomes, the specific gene. So, you know, each person has 23 autosomal chromosomes and then one sex chromosome, one set of sex chromosomes. And then here's what your chromosomes look like, if you guys don't know. So here's your centromere right in the middle, and then you have your P arm, which is the short arm. You have your Q arm, which is the long arm. And each location on this chromosome is a specific gene or loci. Okay, so this is where all your genetic material comes from. And like I mentioned, there's a lot of genetic material on those chromosomes. As you guys all know, the human genome is being sequenced and new findings are produced every day. Okay, so if you think about a specific location for a gene, that's termed the loci, and the locus of the ABO antigen is actually found on chromosome 9. Okay, so that's where your ABO determination comes from, chromosome 9. And then you have the alleles, are the alternate forms of the gene at any given point, okay, on that loci. So you either have your A, your B, or neither being expressed at that gene. That's for ABO determination. And then we have mitosis versus meiosis. Mitosis, the process of somatic cells in which division occurs for cell replication. And the cell must reproduce its chromosomes, so the daughter cell is identical to the parent cell. And this will occur in all your somatic cells except your reproductive. And meiosis is the process for creating a new individual. So you're creating a gamete. It involves your sperm and your ovum. And each carries only one set of chromosomes. So you're inheriting one from each parent. So during meiosis, the process of the gamete formation, the pairs of the chromosomes are going to separate, and all of the separated chromosomes are going to line up at the center of the cell, and then one chromosome from each pair is randomly going to move to either pole of the dividing cell, and then each gamete will inherit half of that chromosomal contents. Okay, you guys all know this. And that'll give you 2 alleles, so for that gene controlling each trait. So given all of that, that gives us infinite possibilities. That's why we're all so different, okay? Sometimes in this process of meiosis, we have what we can call crossover, where the chromosomes line up and then they'll crisscross each other, and that gives you a different gene location. This is not a huge concern for ABO because the ABO's antigens are located very close together, so they usually always inherited together. When we start talking about HLA toward the end of this lecture, HLA, this is a huge concern, and this is why the HLA matching is so complex because of the crossover that occurs, and there's tons of different HLA matches. And we start talking about HLA. If any of you guys have any siblings, your sibling has the better chance of being an HLA match, okay? And the more siblings you have, the better chance. But what's going to happen here is if you have any crossovers as those chromosomes are lining up, notice that if you have the A antigen and the B antigen, they could be at the same loci on 2 different alleles. They could be on the same allele crisscrossed, or they might even be on the same allele. So that's some examples of what happens, and that's why there's so many different variations. And like I mentioned, those genes that are so close together, this is why it's not a big deal in ABO, because the ABO antigens are so close together, they will always be inherited the same. The same is for your M, N, and S. And I thought I had a picture. Here's an example. So on 2 separate alleles, and we start talking about homozygous versus heterozygous, notice that your M and N are always going to be inherited together, and then your S and big S and little s will always be inherited together. They're located very close together, so typically crossover is not a concern for them either. They're always going to be inherited as a haplotype. So when you think about the M, N, and S, they're included in the same family because of their location on the chromosome. So depending upon the position of the genes on the chromosomes, it can affect their expression. So take a look at this example. This is our RH gene, and your RH family is your D, your big C, little c, big E, and little e, okay? So notice the way they could be inherited. You could have D being expressed on one allele, while C and E are being expressed on the other allele. This is what we call trance because they are opposite from each other. When this happens, your trans location will give you a weaker antigenic expression, okay? If your D, C, and E antigen is on the same chromosome, this will give us a stronger reaction. And we start grading our reactions, you'll understand. So the weaker the antigenic expression, the weaker reactions we will have in blood bank, okay? So this one, when it's trans, located on the alleles like this, will give us weaker reactions because the antigen is expressed weaker. Some blood group systems, the antigens might actually be encoded by 2 or more genes on the same chromosome. And then, again, these genes are so close together, they are inherited as a unit, or what we call as a haplotype. And then again, here's your M and N. So your M and N are inherited, and then your S is inherited together. You guys are probably familiar with some of Mendels' laws. The law of independent segregation refers that the transmission of a trait is in a predictable fashion from one generation to the next. And this is why we can kind of use ABO determinations for paternity testing. We can kind of predict what the mom and dad's offspring will be. Each parent donates a gene, and again, that's how we can use it to determine the blood type of the offspring. That's predictable, however, that is not random, okay? It's all genetically determined. So if you have a baby, if you have an offspring that's AB, can one of their parents be O? No. And then you have the law of independent assortment, states inheritance on different chromosomes are expressed separately and discretely. What that means is your RH blood group system that we mentioned in the example, the DC and the little e, those are on one location on chromosome 7, I believe. No, chromosome 1, and then you have your KL group, you'll have big K and little k, KPA and KPB. Those are on chromosome 7. So they're inherited separately. Okay. And then you have your pattern of inheritance. Is it dominant, codominant, or recessive? Actually, very interesting about your ABO blood group, it expresses all of those. So if you think about a AB patient, that's codominant, right? If you think about a O patient, that's a amorph recessive. And then your A and B patients who have the genotype AO, BO, those are dominant. So your ABO group expresses all of that inheritance pattern. And then you guys probably all have heard of Mendels Punnett square using the colored of roses. Notice that your red color is the dominant trait and your recessive trait is the white color of the flowers. So in this example, you have to be heterozygous, meaning that you lack that trait, and so the flowers will be white, okay? Notice that these are dominant, meaning that even if you have the dominant trait and the recessive trait, the flowers are still red, okay? And then you can use frequency, you can determine what the outcome of the offspring would be. So in this example, the offspring has a 75% chance of having red coloration, only 25% of white. So in blood bank, we'll talk a lot about homozygous versus heterozygous, and I hope you guys have a true understanding of what that means. When you start thinking about antigen expression, and we start looking especially at Duffy A and Duffy B, JKA, JKB, M and N, big S, little s. If you have the same expression, let's say both alleles are expressing big S, meaning that on allele 1 you have big S, and then on allele 2 you're also positive for big S, meaning that you're negative for little s. That is homozygous, okay? If you have one of each, if you're big S positive and little s positive, that's heterozygous, okay? And then we're gonna start talking about dosage. If you are heterozygous in your antigenic expression, the weaker antigen expression causes weaker reactions. So it is very common for some of our antigens to demonstrate dosage. And when we start identifying antibodies, we might have some weaker reactions, and you always have to keep dosage in the back of your mind. And we'll go through some examples of dosage, homozygous versus heterozygous. You guys will become very familiar with that. You can actually see homozygous too in our ABO examples. So if both alleles are genetically coded for the A antigen, they will be homozygous, AA, versus heterozygous would be AO, okay? And then for homozygous B, your BB, heterozygous for B would be BO, okay? And then your group O are always gonna be homozygous because they have the A morph, right? They're not expressing either the A or the B. And then, like I mentioned, your genotype is what the actual genetic makeup is. So for AA, that's your genotype, but your phenotype is A, okay? For BB, your phenotype is B. For O, this is your genotype, but your phenotype is O, okay? And then AO, your phenotype is A. AB, your AB, so you have both antigens being expressed. And then BO is a B patient. And like I mentioned, you can have different genotypes, but the same phenotype. So here's an example of that. Your genotype AA, but they still have the same phenotype as a AO patient. Both have the same phenotype. And again, I want you guys to understand with our serological testing, our tube testing that we do in blood bank, we cannot differentiate this, okay? We will only be able to know that they're phenotype A. Here's an example of what I was mentioning about dosage, determining if a antigen is heterozygous or homozygous. And this is what we call an antigram. When we start doing screening cells, you will look at these. And so if you would, this is cell number 1 going across the panel, and cell number 2. These are all of your antigens, all right? So here's your Rh family, D, big C, little c, big E, little e. Here's some very rare antigens as well. We typically don't worry about these. When you start looking at panels, you'll notice all the way down, F and CW will be negative. So typically we won't worry about these. However, in my blood bank experience, we did have a F. We identified an anti-F. So that's your Rh family. Then here's your M, N, and S family. Okay, so M and N, and then big S and little s. Bigger Lewis family, Lewis A and Lewis B. Lutheran, Lutheran A, Lutheran B. Kel, you have your big K, which I think this actually turns Kel1. And then you have your little k, which is called Chelano. And then WA, WB, JKA, and JKB. Okay? So you see how you have the haplotypes. JKA and JKB are a haplotype in the kid family. So when you're looking at homozygous, oh, and one other thing, if there's a plus sign here, that means that cell, so cell number 1 is D positive. That means cell number 1 has the antigen. Okay? So this cell is Rh positive, big C positive, big E negative. Okay? So your zeros indicate that it's negative. All right, so how can we use that? M and N are notorious for showing dosage. Notice that cell number 1 is positive for big M, negative for N. Okay, so it is homozygous for M. Okay, cell number 2 is actually heterozygous, meaning that it's positive for M and positive for N as well. If you take a look, if we were to mix patient plasma, remember that's where our antibody is, and add it to these cells and read for agglutination, we are encouraging that antigen-antibody complex, and we grade our reactions. Notice the cell, here's your reactions right here. Notice the cell that is heterozygous, meaning that it has weaker antigenic expression on the cell membrane, also has a weaker reaction. Okay? If you think about what's going on, heterozygous versus homozygous. Bummer. So here's your red cell, okay? These are your antigen sites all across the cell. For a homozygous, M-positive patient, all of these cells are M, or all of these sites. Okay, those are all M. Heterozygous, can you guys see that? Heterozygous shares the antigen sites, making them weaker. So you would have M and N all across the cell membrane. So there's not as many antigen sites to bind to for your patient's antibody to bind to. That is why heterozygous will give you a weaker reaction. Okay? We have to pay very close attention to that. Weaker reactions in blood bank. You want to make sure, number one, is it due to dosage? Or number two, is it due to multiple antibodies present? Because if there's different antibodies, they might react at different strengths. Okay, so that's why your reactions are so important, and that's how dosage works. And you can look at M and N are notorious for dosage. Your KID family is also notorious for dosage. So we could actually take this example and demonstrate dosage with your KID family. Notice that cell number 1 is homozygous, positive for JKA, negative for JKB. Cell number 2 is positive for both JKA and JKB. That would give us a weaker reaction in patient plasma. Does that make sense? So JKA is notorious for dosage. Sometimes you might see dosage with some of your RH family. It's not as common. They usually react pretty strong. So.
I was just wondering, when you say antibody reaction, is that from the patient's plasma?
Yes.
Itself?
So you're taking patient plasma where your antibody is, and you're seeing if it reacts with any of these cells. These cells have a known antigenic profile, and so then we can kind of see what's positive on cell number 1 and 2 that's given us these reactions. And then this is what we call, this would be like an IAT, and so this is what your antibody screen looks like.
So this would be a reverse typing?
Yeah, it's a form of reverse typing because you're using patient plasma. And we are seeing what antibody is present in your patient plasma. But the antigenic expression of each cell is very important in determining homozygous versus heterozygous, and we would look at our reactions. Does that make sense to everybody? So in my demonstration, when I did, homozygous versus heterozygous, you have to think about the number of antigen sites present. So if you're homozygous, you're going to have both M and N antigen sites on that cell. So there's fewer number of antigen binding sites. If that patient had anti-M, it can't bind as many times on that cell. So your reaction is going to be weaker, okay, if it's heterozygous. And again, when we're doing our serological testing, the genes might not be detected, so if we can tell a patient is A, but we don't know if they're AO or AA, with our serological testing, we just can't do that. However, if you look at our haplotypes that travel together, N and N, this does give us their genotype, right? We know that cell number 2, this patient, whoever donated this cell for testing, was M positive, so they are heterozygous. So we can get that genotype. We know that. And then all of your other big S and little s, Lewis A and Lewis B. Lewis A and Lewis B, you're either positive for one or the other, and you can be negative for both, but you cannot be positive for both. We'll talk about the Lewis family a little bit later. And then your KEL, your Duffy A, Duffy B, and JKA and JKB. Those are your haplotypes. They're inherited together. So if you can detect the gene only in homozygous, then of course the trait is recessive. You guys know that. An example of that is our group O, like I mentioned. They have to have the amorph gene, OO. So if your heterozygous alleles are both expressed, like such as our group AB, then that is codominant. An example of that, if you did Mendels studies with the color of the flowers, an example of that would be if you had one dominant gene and one recessive gene, then the flowers would be pink, and that's a codominant. And like I mentioned, that group O is called an amorph. It produces no visible effect. So that A antigen or the B antigen is not being expressed. And then if it's related to the sex chromosomes, you guys all know we call it sex-linked. If it's only located on the X chromosome and the Y chromosome, we do have some red cell antigens that are sex-linked. One of them is XGA, and it is not included on this panel. Sometimes you'll see it on panels and sometimes you won't. So here's an example of your Punnett square using the codominant. So both the homozygous and the heterozygous. So if you have the dominant gene and the recessive gene, then your flowers are going to be pink. So if you notice, you have a 50% chance of flowers being pink and a 25% chance of your flowers being red and 25% chance of your flowers being white. If both of your parents are heterozygous, right? So your phenotype is the observable expression of inherited traits. So like our patients that are A, their phenotype is A, their genotype would be AO or AA. We can use this to predict paternity studies. It used to be used quite a bit, the ABO of mom and dad, to determine if a father was indeed the father. But now it's more molecular based. Now they do paternity testing, DNA. Okay, as you guys learned in the previous lecture, the blood group antigens are embedded on the cell protein. They're actually derived from protein synthesis will determine how those antigens are expressed on the cell. And you guys know DNA, we describe it, helix DNA, coiled. It's described as like a string of beads or a string of pearls. And then the DNA within the gene is what contains the genetic information. So if you think about what composes that DNA molecule, each nucleotide is composed of a sugar, a phosphate molecule, and then a nitrogenous base. And then your 4 nitrogenous bases in DNA, you guys all know this. This is just a review. You have your purines and your pyrimidines, adenine and guanine being your purines, and thymine and cytosine being your pyrimidines. So adenine and thymine must pair. Guanine and cytosine must pair, okay? And this is occurring in your cell nucleus. Okay? This is where your DNA is. So what's going to happen is that DNA is located in the nucleus. It's going to unfold. Messenger RNA is going to come in, and it's going to kind of match up with your DNA, and that messenger RNA is then going to travel to the ribosomes of the cytoplasm and allow for protein synthesis. And this schematic just demonstrates what's going on. So we're in your nucleus right here. Here's your double-stranded DNA. And then here's your messenger RNA. So your DNA is going to uncoil. Your messenger RNA is going to come in. It's kind of like a pattern. And it's going to match up. And then your messenger RNA is going to travel to the ribosomes. Transfer RNA is going to bring in your amino acids. And your amino acids will form this polypeptide chain. That polypeptide chain of amino acids is what will determine your antigen expression on the cell membrane. That's a review for everybody, right? If you look in this example, notice that each transfer RNA has 3 proteins there, so that's your codon. Codons will determine the pattern of your proteins. Some of those codons determine when it stops and starts as well, so you'll have start codons and stop codons. So just like we talked about in hematology, where we have some abnormal hemoglobin synthesis, any amino acid rearrangement in that structure could cause a different antigenic expression on the cell as well. So we've talked about how ABO and antigenic expression is genetically linked, and we can use that to determine how it will be inherited in families. And we can do that through what we call a pedigree. I don't know if any of you guys have ever done pedigree studies before. And then we can also do the Punnett square. So here's an example of a pedigree where you take the phenotype of the parents, possible genotypes, and then we can determine what will be the possible phenotype and genotype of their offspring. Okay, so in this example, one parent is A with a genotype of AO, the other parent is B with a genotype of BO, and then here's all of the possibilities. They could have an AB patient, they could have a homozygous or heterozygous A, meaning that AO or BO, or they even could have an O patient. Notice in the pedigree and in the other example, with an AB patient, even though one parent is AB, one parent is O, notice that they do not have the capability to have an O offspring. And that's just because, remember, in order to be O, you have to have both the O gene expression on both alleles, okay? And remember, you can only be OO to be O. So in order for a patient to be O, they have to have one O allele from one parent and one O allele from the other parent. So if anybody ever tells you that they're AB and their mom is O, might want to check that out. All right, so let's do some practice, Punnett squares. You might see this on board of certification exam, being able to determine what the probability of the offspring is based on parents' ABO. And I'm sure you guys all know this. So if one parent is, mom is AB and dad is O, what's our potential blood types? AO, BO. Can you have a O? No. So we have 50% chance that the offspring will be type A, 50% chance that the offspring will be type B. Mom is AA and dad is BO. Can they have an AB offspring? Can they have an O offspring? No. So our options are AB, AB, AO, and AO. So their offspring will either be AB or A. What if mom is B and dad is A? Both of them are heterozygous. They can't have an AB. Can they have an O? Yes. And they can have an A and they can have a B. So 25% of each. And then here's some examples of various combinations of mom and dad with the potential offsprings. So that's pretty easy. And I feel like that was a review for everybody. The thing I want you guys to remember when we start getting into our RH family, this will be very important. How your RH are inherited. Notice that your D, C, and E, they're all traveled together. And so when we start talking about RH, we'll come back to this, okay? All right, any questions? I finished that lecture kind of early.
Way back.